Activated macrophages secrete a range of cytokines that have a variety of local and distant effects. Cytokines are small proteins that are released by various cells in the body, usually in response to an activating stimulus, and induce responses through binding to specific receptors. They can act in an autocrine manner, affecting the behavior of the cell that releases the cytokine, or in a paracrine manner, affecting the behavior of adjacent cells. Some cytokines can act in an endocrine manner, affecting the behavior of distant cells, although this depends on their ability to enter the circulation and on their half-life. Chemokines are a class of cytokines that have chemoattractant properties, inducing cells with the appropriate receptors to migrate toward the source of the chemokine. The cytokines secreted by macrophages in response to pathogens are a structurally diverse group of molecules and include interleukin-1, IL-1, interleukin-6, IL-6, interleukin-12, IL-12, TNF-alpha, and the chemokine interleukin-8, IL-8. The name interleukin, IL, followed by a number, for example IL-1, IL-2, and so on, was coined in an attempt to develop a standardized nomenclature for molecules secreted by and acting on leukocytes. However, this became confusing when an ever-increasing number of cytokines with diverse origins, structures, and effects were discovered. And although the IL designation is still used, it is hoped that eventually a nomenclature based on cytokine structure will be developed. The cytokines and their receptors are grouped according to their structure. There are three major structural families. The hematopoietin family, which includes growth hormones as well as many interleukins with roles in both adaptive and innate immunity. The TNF family, which functions in both innate and adaptive immunity and includes some membrane-bound members, and the chemokine family. Of the macrophage-derived interleukins, IL-6 belongs to the large family of hematopoietins. TNF-alpha is obviously part of the TNF family, while IL-1 and IL-12 are structurally distinct. All have important local and systemic effects that contribute to both innate and adaptive immunity. The recognition of different classes of pathogen may involve signaling through distinct receptors and result in some variation in the cytokines induced. The study of this is still in its infancy, but it is thought to be a way in which appropriate responses can be selectively activated as the released cytokines orchestrate the next phase of host defense. And that's activated macrophages secrete a range of cytokines that have a variety of local and distant effects. Have a great day.